So we're from Tapton. I'm Rebecca, this is Anthony, and this is Sophia, and we've been working in partnership with Sheffield University to do this work on the motor neuron disease.
So motor neuron disease is a fatal neurodegenerative disease which affects the motor neurons, so the messages that your brain sends to your muscles to tell you where your hands go and how to breathe and that kind of thing. So we don't really know let's go here. Right, we're good now. We don't really know what causes motor neuron disease, the underlying mechanisms, but there are definitely loads of theories about it. And the main one being at the moment is this big yellow dot here, which is a gene called C9orf72. And as you can see the graph there, this is like all the research that's been going on over the years. So C9orf72 is a gene that we think is responsible for creating these toxic dipeptide repeats, which are basically like little proteins made of 2 amino acids that accumulate in the cells of the motor neurons, and then when these build up, it makes the neuron degrade. So Angeline will tell you more about the mechanism behind all this, but basically there's a gene which has a repeat sequence of 4 Gs and 2 Cs, which is falsely exported out of the cell. So when you're translating DNA to make proteins, it goes from pre-mRNA and then it's spliced to mRNA, and then but basically this doesn't happen, and we're trying to work out why it's transported before it's been properly packaged.
So basically, nuclear export proteins are being sequenced by the G4C2 repeat sequence, and this repeat sequence is being falsely exported because it's still pre-mRNA and hasn't been spliced yet. So this means that we have RNA being translated by the ribosome, which shouldn't be turned into proteins. And this then, in this repeat sequence, you can produce 6 dipeptide repeats, and these then aggregate in the cell and sequester the important cellular factors, which then causes neuron degeneration. And scientists have found the protein SRSF1 to be involved in the false exportation of pre-mRNA, as it binds to the unspliced mRNA. And we've done a test on the Drosophila fruit fly and found that if you remove the protein SRSF1, you save the phenotype and prevent degradation, even if you have the repeat sequence. So basically, it doesn't matter if you have the repeat sequence or however many you have. If you can get rid of the proteins, then you can prevent the degeneration.
Okay, so what we've actually been doing as a school is we've been trying to splice in these repeat sequences, the G4C2, into yeast using plasmid, yeast expression vectors. And then we're looking for the toxic phenotype, which is associated with motor neuron disease. And also it was in the fruit fly's eyes. You can bring might be the next slide. But or not. So then what we're doing is we're then going to, using the yeast deleted gene library, we can find all the nuclear export factor genes, and we can get them deleted, and then we can see if then the phenotype is then rescued. And then what we're hoping for is that the same nuclear export factors would be found in human cells, and then we could look for potential targets for drugs, because our aim is to find the specific nuclear export factors that are involved in bringing out this repeat sequence to it. Thank you.